This is the part 2 of our evaluation exam discussion for biochemistry. Or remember, let's pick up where we left off. So we are in item 41. But before that, how's your score so far? Sir, pasa naman. Sir, kumakapit. Class, again, remember our passing score is 56 out of 80. So I do hope that you are getting good scores and correct answers, diba? So without further ado, let's start again our discussion with item number 41. All right, let's read. The rate of oxygen consumption is increased upon addition of NADH because, class, be patient in reading your choices. Let's start with letter D. NADH uncouples oxidation from phosphorylation. Class, clearly this is an incorrect choice, okay? See, in our electron transport chain, of course, we use your oxidative phosphorylation to power the production of ATP. So it does not uncouple it, all right? Also, letter C. NADH is an allosteric activator of NADH ubiquinone oxidoreductase. Class, again, your NADH ubiquinone oxidoreductase is the enzyme that catalyzes that guides your electron transfer in complex 1. But of course, it does not explain the oxygen consumption increase in your ETC, okay? So again, may better choice tayo aside from letter C. Let's check letter B. Cytochrome oxidase binds NADH and transfers electrons from NADH to oxygen. Class, this is an incorrect choice. Sir, bakit? Hindi po direct ang electron transfer to your NADH to O2 using only one complex or enzyme, which is cytochrome oxidase. So, sir, letter A ang answer? Yes po. Again, the oxygen use increases as NADH donates electrons to our electron transfer chain, the four complexes, before ending with oxygen as the final acceptor. Let's read letter A. Electrons from oxidation of NADH are transferred through electron transfer chain to your oxygen. So, class, again, our answer here is letter A. All clear? Ayan, I hope you got the correct answer. So again, ang challenge here is the lengthy choices. Class, before we discuss our next two questions, let's have a recap first of your citric acid cycle. Class, remember the citric acid cycle, sir, unique yan because again, it is amphibolic. But of course, we highlighted its catabolic function in order to oxidize your fuel molecule in the form of carbs, lipids, and amino acids. Natatandaan pa bang energy products? Ayan, you have your GTP or also your ATP, they are just equivalent to each other, and your NADH and FADH2 as the electron carriers. For the location, remember, mitochondria, diba? And for requirement, strictly aerobic. And here is the overall reaction. Again, our initial substrate is acetyl-CoA to be condensed to our initiator molecule, oxaloacetate. Alright, so class, let's proceed with our next two items, sorry. Which of the following statements regarding Krebs cycle is true? Sir, parang nasa practice test yan. Let's check. Of course, you already know the answer here. So letter A, it occurs daw in the cytosol. No, again, mitochondria po. Letter B, amphibolic. Yes, that's the correct answer. But let's check the other two choices. Letter C, it is an anaerobic process. No, again, strictly aerobic po, si Krebs cycle. And lastly, it contains no intermediates for gluconeogenesis. Class, remember your TCA cycle intermediates, diba? So your gluconeogenic pathway has a similarity in terms of their intermediate, and that is your oxaloacetate, diba? Your oxaloacetate is generated in your gluconeogenic pathway. Alright, so class for 42, answer is clearly letter B. Okay, so for our next item, class, I hope you are familiar with our electron carriers in our ETC. So which of the following electron carriers is not, class, careful is not a component of the mitochondrial electron transport chain? Again, we know that nicotinamide adenine dinucleotide is one of our electron carriers. We fit it in your complex 1. So this is an incorrect answer, diba? So again, it's a part of your ETC. Sir, si coenzyme Q po, di ba mobile electron carrier yan? Yes, that's why it's also incorrect. Si letter D, flavin mononucleotide. Sir, parang hindi natin na-discuss yan. Class, remember, that's a coenzyme of your complex 1, di ba? So it guides your electron transfer that's occurring in your complex 1. And remember, si NADP or your nicotinamide adenine dinucleotide phosphate is on your biosynthetic pathways. It's not in your electron transport chain, di ba? So I emphasize this in our discussion sa pentose phosphate pathway. Alright, for 43 again, answer is letter B, NADP or nicotinamide adenine dinucleotide phosphate. The phosphorylated form ni NAD+. Ayan, so class, ready na with our next item? Okay, so 44, what additional reactant is required for oxidation of PUFAs or polyunsaturated fatty acids compared to your saturated fatty acids? Class, recall, here are the choices. A, biotin, B, NADPH, C, oxygen, and D, ATP. Class, sir, na-discuss ba natin si biotin sa beta oxidation? No po. Dapat familiar kayo again, we do not use this in beta oxidation as our coenzyme because its main role is for carrying your carbon dioxide in carboxylation reaction, such as in GNG or gluconeogenesis. Diba? Next, NADPH. Again, this is used in our PUFAS oxidation. Sir, where specifically as reducing agent for double bond rearrangement. So class, again, take note. NADPH can also participate in PUFAS oxidation to initially reduce your double bond or serve as reducing equivalent for rearrangement. Alright, thirdly, oxygen, final electron acceptor siya ng electron transport chain. So no direct connection in your beta oxidation. And lastly, sa ATP, either your PUFA and your saturated fatty acids use your ATP as their energy source. Okay? So because they activate, your ATP activates your fatty acids in our activation step, diba? So we discussed it. Particularly, we use 2 ATPs pa in order to fully activate your fatty acids. So class, again, for 44, answer is letter B, NADPH. Ayan, so I do hope you got the correct answer. Again, your NADPH participates in PUFAS oxidation as source of reducing equivalents for double bond rearrangement. Alright, so let's proceed with our next two items. So this is still under your metabolic pathways in general. Class, for this question, I emphasize this in our crash course discussion. So, di ba, we prioritize a certain biomolecule as our main energy source. Which of the following fuel reserves can be used by skeletal muscle? A, protein, B, glycogen, C, fatty acids, D, all of the above. Ay, sir, may all of the above, letter D ang answer, 100% confident. Let's check. Again, class, diba, we discussed that these three are all fuel molecules from our discussion, diba? So carbs, lipids, and amino acids can serve as energy source. And remember, their prioritization again, first glycogen, then fatty acids, then protein. And thus, all of them can be used by your skeletal muscle, depending on what is available for them. Okay? So for 45, answer is letter D. Alright, so class, 4 to 6. Sir, challenging, hindi natin na-discuss. Yes, class, but this is still under your beta oxidation, the side reaction of your beta oxidation. Let's check. Which pathway leads to the formation of dicarboxylic acids as an end product? Is it A, beta oxidation, B, alpha oxidation, C, pentose phosphate pathway, or D, omega oxidation? O class, clearly, our beta oxidation is not the correct answer, okay? So this is, in our beta oxidation, our product is acetyl-CoA. It's not a dicarboxylic acid, all right? Sa pentose phosphate pathway, remember, our products are sugars and NADPH. Oh, it's just between your letter B and letter D. Oh, diba? Through the process of elimination, you got now a 50% chance of getting the correct answer. Oh, class, palakasan ng guardian angel. What is your answer? I know that we did not discuss this because, again, this one is an advanced topic. For 46, our answer is letter class, I do hope you got the correct answer. Letter D, omega oxidation. Sorry. Oh, let's discuss briefly your omega oxidation. See, this is an alternative pathway other than your beta oxidation. So class, this is triggered or activated when our beta oxidation is impaired. So meaning, pwedeng may lack of enzymes or may defect ang enzymes that participate in beta oxidation. For instance, we have your glutaric acid. So class, your omega oxidation, actually, this process, of course, ang first one is the hydroxylation of your fatty acid, and hence it will form its hydroxy derivative. Sir, do we need to study the specifics? No po. So we summarize again. Here kasi we call it your omega oxidation since the target carbon is the last carbon in the chain. Okay? So ang location yan mostly sa ER, sa endoplasmic reticulum. And again, ang common modification is through hydroxylation in order to convert it to dicarboxylic acid via another round of oxidation. So meaning, si OH upon addition, i-oxidize siya to form another carboxylic acid. That's why we have now a dicarboxylic acid functional group. And ang outcome, ayan, it actually increases water solubility for excretion or for other pathways use. Alright, so again, omega oxidation, main modification, hydroxylation, and oxidation to form dicarboxylic acid as end products. Again, no need to remember the specific mechanism. Alright, so class, I do hope tumama kayo sa guess nyo, diba? So for now, let's proceed with our next item. Oh, class, this one, very lengthy din ang choices. So this is actually about your hormonal regulation. Let's see. Glucagon, which of the following statements is false? Let's read. A. Glucagon increases cyclic AMP levels in the liver in response to the fasting state. Class, remember, this one is true. Bakit nga ulit? During fasting state, our glucose level in the blood is really low. So that's why we will release your glucagon. Again, glucagon meaning glucose is gone, that's why it should be activated. And the AMP, of course, that's your signal in our body, particularly cyclic AMP signals our hormone expression that our body is in low blood glucose level concentration. That's why letter A is a true statement. This is not the false statement that we are looking for. Let's check letter B. Epinephrine causes mobilization of tags from adipose tissue in response to stress. Yes po. So class, when we are stressed, our body finds alternative energy source, because they do not want to deplete our glycogen reserves. Okay? That's why we use your fight or flight hormone, diba? Si epinephrine in order to mobilize our tags as our source of energy during stress. Ayan. So class, letter B again, true statement, not our answer for 47. Let's check letter C. Insulin decreases gluconeogenesis in the liver and increases glucose uptake and glycolysis in muscle. Sir, true ba yan? Yes po. Remember, ang main function ng insulin is to mobilize your glucose. So in order to mobilize your glucose, of course, meron tayong high blood glucose level. So hindi natin need ang gluconeogenesis because again, this is production of glucose. Okay? But we have high blood glucose concentration. That's why it needs to increase the glucose uptake and the usage of glucose through glycolysis. Okay? Not to produce your glucose. So obviously, letter D is our answer for 47 because your glycogen phosphorylase can be affected by insulin and glucagon, not exclusively insulin lang class. Okay? So to sum up, for glucagon, again, target tissue, liver only. So it can increase the expression of your cyclic AMP in order to produce glucose. All right. So again, here you have an increased expression of gluconeogenesis and glycogenolysis. Again, when you have glucagon, meaning your glucose is gone. Glucose is gone, diba? Second, si epinephrine for fight and flight response. That is our responsible hormone. So we have three target tissues, particularly in muscle and adipose. It mobilizes your fatty acids primarily. So si glucose, of course, that's heavily mobilized in the liver. All right. Letter at number 3. Insulin, liver, muscle, and adipose din class. But here, you see, it can lower the expression of GNG because again, we do not want glucose. We actually need to decrease the blood concentration of the glucose. Diba? So again, it will increase glucose uptake and increase glycogenesis because we have an excess of glucose in the blood. And lastly, si glycogen phosphorylase, its target tissues are liver and muscle. And of course, class, again, it can be both affected by glucagon by activating it and insulin by inhibiting it. Okay, so class, again, our answer for 47 is letter D, your glycogen phosphorylase hormonal regulation. All right, let's proceed with our next item. Which of the following is a direct product of the oxidative phase of the pentose phosphate pathway? Our choices, ayan, A, ribose 5-phosphate, B, fructose 6-phosphate, D, C, NADPH, or D, glyceraldehyde 3-phosphate. Sir, ano ba yung answer dyan? Oxidative, diba? Hindi po ba reducing equivalent? Yes, class, that's correct, particularly si NADPH. Okay? Again, class, ang sugars, such as your A, B, and D, they are generated in non-oxidative phase. Diba? Recall, for oxidative phase, our energy yield is 2 NADPH per glucose 6-phosphate consumed. Diba? Very easy si 48 if you know the ins and outs of your PPP. Alright, 48 is letter C. 49. Sir, parang hindi natin na-discuss yan. Nahula na lang ako. It's okay, class. So let's check your knowledge. Again, very trivia-based. C, biochem. In mammals, beta oxidation occurs in both mitochondria and your peroxisomes. However, in plants and most fungi, the beta oxidation cycle occurs primarily in that certain organelle we call as BLOM. A, glyoxysomes, B, lysosomes, C, peroxisomes, or D, endoplasmic reticulum. Class, what is your answer here? So of course, hindi na siya peroxisome, okay? Because it's already stated in the question. So impossible na peroxisomes yan, okay? So now we are left with 3 choices. And of course, class, C. Ayan, for your plants, ang main site ng beta oxidation includes your mitochondria and your glyoxysomes, a type of peroxisomes, okay? So class, again, heavily localized or concentrated ang beta oxidation in plants in your glyoxysomes. Who got the correct answer? Okay, very good, class. Again, please remember, your plants have specific and unique organelles that are solely for beta oxidation, and that's your glyoxysomes. Okay, so for 49 again, letter A. Sir, ang challenging yan. Yes, because again, we did not discuss it. O class, again, it should be noted that our topics in crash courses are all core concepts lang. Para hindi kayo ma-overwhelm. But don't worry, again, we discussed naman each concepts in our items here in our evaluation exam discussion. Okay, so class, let's proceed with our item number 50. The product of the Calvin cycle that is used to replenish ribulose 1,5 bisphosphate is, class, again, this is also your initiator molecule, diba? Si ribulose 1,5 bisphosphate. And in order to replenish this molecule, what do we need? What specific reagent? Is it letter A, sorry, ADP, B, G3P or glyceraldehyde 3-phosphate, C, NADP+, or letter D, ATP? Class, naaalala bang specific reaction? Ayan, so as you can see, in order to regenerate your R15P bisphosphate, sorry, you need 2 molecules ng glyceraldehyde 3-phosphate. Okay, so this is through a condensation reaction. O sir, ba't hindi sa ATP? Any din yan. Okay, check and read and comprehend the question carefully. Product daw po ng Calvin cycle also. So we can limit actually our answer here to letter B. Again, this is our direct product coming from your Calvin cycle. Alright, again, for number 50, our answer is letter B, glyceraldehyde 3-phosphate. Easy ba ang number 50? Yes sir, basta alam po ang substrates. O diba? So let's proceed, class, to our next item. 51. Triglycerides contain more metabolic energy per unit weight than glycogen because, let's check. Letter A, the carbohydrate glycerol is readily metabolized. Is this correct, class? So actually, this is an incorrect statement, okay? So again, si glycerol is a backbone of your triglycerides, not your carbohydrate, okay? So I hope that's clear. This is an incorrect answer. Letter B, anhydrous glycogen cannot be cleaved by glycogen phosphorylase. Class, either anhydrous and your hydrous glycogen, we use it in our biological system because your glycogen phosphorylase can both cleave or degrade them when we need your glucose, okay? So again, this is not your correct answer. Let's go to letter D. Hi sir, letter C ang answer, no? I don't know. So again, let's proceed with letter D muna. Triglycerides have a higher average molecular mass. So class, your molecular weight does not affect the metabolic energy yield of each biomolecules, okay? So actually, ang main factor that can affect the energy yield is the structure of your molecule, if it's readily reduced or if it's heavily reduced rather, or somehow oxidized, okay? And that is letter C. So remember, si fatty acids, because of its chain of hydrocarbons in its structure, that's the reduced form or the most reduced form comparing it to your carbohydrates. So letter C, of course, is our answer here. Again, fatty acids are less oxidized than carbohydrates. Let's see and check the comparison here. For fatty acid, it is highly reduced, meaning there are more electrons released ready for your electron transport chain. O di ba, class, remember our discussion on ATPL calculation sa lipid metabolism, comparing it to your cellular respiration, wherein we use your carbohydrate, particularly glucose. Of course, large amounts of energy is coming or are coming from your fatty acid metabolism. Okay, because carbohydrates, they are more oxidized, so meaning fewer electrons na lang ang capable nilang i-give up or i-release. Okay, so again, in terms of your hydration, it does not really affect the metabolic energy yield. Okay, so again, energy content, ayan, just a supplementary. For fatty acid, around 9 kcal per grams. Comparing it to carbs, ayan, we have a value 4 kcal per grams lang, class. Okay, so again, for number 51, our answer is letter C. Alright, so class, for our next 3 items, let's first revisit the summary of our photosynthesis discussion. So again, remember, for your light-dependent and light-independent, they are both occurring in your chloroplast. In particular, si light-dependent in your thylakoid, si light-independent again, or your dark reactions, or your Calvin cycle, or your C3T, C3 photosynthesis, ang dami niyang alias, di ba? It's in your stroma. Or recall, love trip, dog show, di ba? Light-dependent, thylakoid, dark reactions, stroma. In addition, for our reactants again, ang main reactant for light-dependent, of course, light and water. And we have your coenzymes, NADP+, and ADP. On the other hand, for light-independent, of course, we have an input of your carbon dioxide, and we will use up the ATP and NADPH generated from the light-dependent reaction, okay? For the products, ayan class, again, recall the energy products that were used in light-independent as well as our product of water-splitting reaction, si oxygen. For light-independent, that's just sugar intermediates. Again class, ang purpose ng photosynthesis in general is to produce sugar molecules. For regulation, again, ang light intensity, direct ang relationship with light-dependent. The more light is available for our enzymes in your light-dependent, of course, the more readily they can excite the electrons. For light-independent, the energy substrate availability po ang for regulation discussion natin. Okay, so class, don't also forget your photorespiration process. So again class, unwanted po ang photorespiration ha. That's why our plants adopted different mechanism, diba? Si C4 and si CAM pathways. For photorespiration location, it can be done in your stroma, peroxisome, and mitochondria. And again, instead of carbon dioxide, we actually use oxygen rather than producing it. And for the products, ayan, instead of being a reactant, si carbon dioxide, is actually a product of photorespiration. And again, this occurs during low carbon dioxide level or high oxygen. Again, low CO2 or high oxygen in the environment. So this happens under hot and dry conditions. Already na class, this is just a brief summary of your photosynthetic pathway. Okay, so let's proceed with our next item. Number 52. Which of the following statements about photosynthesis is false? Let's see. Let's start with letter D, okay? So the light reactions occur within or on the thylakoid membrane. So class, is this true? Yes po. Diba? Love trip, dog show. Light reactions in your thylakoid and si dark reaction sa stroma. Also letter C, yun, discuss mo na. Yes po. Si letter C is also a correct answer. Okay, so again, this is a true statement. That's why this is not our false statement that we are looking for. Let's check letter B. The light reactions oxidize water to oxygen. Yes, that's correct. Reducing NADP+ to produce NADPH in the process. Also correct. Class, I do hope that you now know the specifics of our light-dependent reactions and dark reactions. So lastly, in plants, much of the carbohydrate form is stored as sucrose. Class, this is an incorrect statement. Again, ang main energy storage ng plants ay starch. Okay? So again, A is a false statement and thus the correct answer. For here, class, again, remember, laugh trip, dog show. Ayan, so I do hope na magamit yung mnemonics natin. Let's proceed with our last 2 items for your photosynthesis. Photorespiration happens when, class, diba, as we have discussed earlier in the summary, low carbon dioxide and high oxygen. Let's check. Water is limited. Wala pong connect yan. Okay? So it's just either your carbon dioxide that is low or your oxygen that is high in concentration. O CO2 is high. So of course, favored ang carboxylase reaction ni Rubisco. So this is an incorrect answer. O si letter C, oxygen is low daw. So class, this is not the correct answer. Actually, our answer here is letter D. Class, for this type of question, if you see contradicting choices, either one of them lang ang correct. O diba? Si CO2 is high, si CO2 is low. So make it make sense. So either one of them lang ang correct answer. And that is letter D, carbon dioxide is low. O clear? Yes sir, very easy. Si number 53. O let's check number 54 kung easy din siya. In Calvin cycle, the following enzymes are involved except blank. A. Phosphoglycerate kinase. Yes po. It catalyzes our production or phosphorylation ng phosphoglycerate. Okay? Letter B. Ferredoxin NADP plus reductase. Sir, parang nasa light reactions yan to generate your NADPH. Yes po. Okay? So again, letter B is actually our correct answer. But let's check the remaining two choices. So glyceryl dihydrophosphate dehydrogenase. Yes, that's in your Calvin cycle in order to generate our initiator molecule, ribulose 1,5 bisphosphate. And lastly, of course, Rubisco, the first enzyme that catalyzes the first reaction or the carbon fixation reaction. Okay, for number 54, sorry, answer is letter B, ferredoxin NADP plus reductase. Class, before we proceed, kamusta ang scores? Sir, barely breathing. Bakit class? Again, I hope na sana after your discussion here, dapat may strategy kayo. Okay? So of course, item analysis muna. What are your weakest points? Sa carbs ba? Lipids? Or sa cellular respiration? Or sa photosynthesis? Okay class, dapat you are studying smart. Class, in relation to number 54, again, recall your non-cyclic photophosphorylation. So it is involved in your non-cyclic photophosphorylation and not in your Calvin cycle, okay? So this is your ferredoxin NADP plus reductase complex. So they are responsible in the electron transfer all the way through production of your NADPH, okay? Again, in NCP, we both use your photosystems 2 and 1, and here's the specific electron flow in order to produce your NADPH reducing equivalent. Okay, so class, that's it for photosynthesis. Now let's proceed with our next item, item number 55. So what is the main source of glucose carbons for gluconeogenesis? Class, here are our options. Is it A, guanine, B, alanine, C, cysteine, or D, threonine? Sir, parang naligaw si guanine. Is it a nucleic acid? Yes, so this is not actually our correct answer. So it's just about letter B, C, D. And all of them are amino acid. Class, recall, we discussed this in our transamination reaction, diba? Particularly in the muscle. That is your glucose-alanine cycle, diba? So of course, the alanine is the main source of glucose carbons for gluconeogenesis. Remember, in our muscles, of course, glycolytic pathway will occur. That's why, ang result, ang key product dyan is a pyruvate that will be converted to alanine in order to transport it to your liver in order to convert it back to glucose via gluconeogenesis. Okay? For number 55, our answer is letter B, alanine through the glucose-alanine cycle. Clear? I hope you got the correct answer, class. Very easy po ang number 55 or 56. During the gluconeogenic conversion of pyruvate into glucose in the liver, all of the following enzymes are involved except, class, simply ang ina-ask niya lang which of the following is not a bypass enzyme in your gluconeogenic pathway. Let's check. So of course, letter A, pyruvate carboxylase. Yes, sir. That's the first bypass enzyme. Tama. So letter D, glucose 6-phosphatase. That's the last bypass enzyme, diba? So it's either letter B and letter C. And class, remember, we don't have pep carboxylase. Okay? It's pep carboxykinase that catalyzes the conversion of your oxaloacetate to phosphoenol pyruvate. Okay? So our answer here, letter B, of course, hindi siya involved in your gluconeogenic pathway. Alright, number 56, our answer is letter B. Or class, remember, again, our gluconeogenic pathway and the bypass reactions. We have pyruvate carboxylase and pep carboxykinase for the first glycolytic enzyme bypass reaction. We have your fructose 1,6-bisphosphatase, the committed step of gluconeogenesis, and of course, your bypass enzyme here in your glycolytic pathway is PFK1. And lastly, glucose 6-phosphatase to bypass the hexokinase reaction. Okay, so again class, ayan ang reactions in your gluconeogenic pathway. They are thermodynamically irreversible. Clear? Ayan, can we proceed with our next item? Konti na lang class. We are now in our item number 57. So what or which is the origin of the majority of the ATP used in gluconeogenesis? Class, I will rephrase the question. Which of the following pathways in the choices produces large amounts of ATP? Let's check, diba? That's your tags. And which among the choices are about your lipid beta oxidation? That is letter A. Okay? So again class, ang breakdown of amino acid, list priority siya in energy production because relatively low ang energy yield with your amino acid. Because again, our product is just glycolytic intermediates, diba? Your carbon skeletons will be used for TCA cycle intermediates replenishment. Also, sir, si C and D, wrong din yan because they are carbohydrates. Yes po. Again, relatively large amounts ng energy ang nafo-form ng beta oxidation. And that's why majority of our ATP are coming from fatty acid beta oxidation, particularly for gluconeogenesis pathway. O, diba? So again, for breakdown of amino acid, carbon skeletons ang product. For degradation, glucose. So it will be fed to cellular respiration. And remember, our yield in your cellular respiration is relatively lower or really significantly lower compared to your fatty acid beta oxidation, diba? Around 30 to 32 ATP per glucose. And lastly, for oxidation fructose 6-phosphate, similar reason class with your degradation ng glycogen. Clear? So for 58, ayan. O sir, parang did kino segment yan during our discussion. Yes po, diba? I emphasize this. What is the intermediate that links the urea cycle and TCA cycle? O, diba? I have a did kino segment there. Is it A, fumarate, B, succinate, C, citrulline, or D, argininosuccinate? Sir, I can recall that's a TCA cycle intermediate. So can we eliminate letter C and letter D? Yes po. So it's either letter A or letter B. And clearly class, I know this is very easy for you, diba? We have your aspartate argininosuccinate shuttle, remember? And our linking TCA cycle intermediate there is fumarate. O, don't be confused again ha. Ang linking intermediate natin is a TCA cycle intermediate fumarate. O, diba? Again, your aspartate argininosuccination is a bridge that connects your TCA and urea cycle through your fumarate intermediate in the TCA cycle. So again, this mechanism makes efficient nitrogen and carbon flow pathway or flow between the two pathways. Clear? Alright, so class, let's proceed with our item number 59. O sir, medyo na-confused ako. Parang lahat kasi ng answers pwede. Let's check. All amino acids pass through A. Glycolytic pathway and PPP. B. Citric acid cycle and PPP. C. Glycolytic and citric acid. Or D. Glycolytic, PPP, and citric acid. Class, for these pathways, si PPP po ay not included in our amino acid metabolism. Okay? So again, it does not participate in amino acid metabolism because its primary function is to produce an ADPH and ribose 5 phosphate. So not in the main energy generating or gluconeogenic routes where your amino acids are used to break down or to be degraded. Okay? So again, si letter A because may PPP siya, si letter B and si letter D are incorrect choices. Class again hindi po nagpa-participate si pentose phosphate pathway in amino acid degradation. So sir, C ang answer? Yes po. Again, only your glycolytic pathway and citric acid cycle are being used in our amino acid metabolism. In particular, check nyo, all amino acids are funneled through the intermediates of your glycolytic pathway or citric acid cycle. Diba? We have your gluconeogenic and your ketogenic amino acid. Alright, so class again, that's just your glycolytic and citric acid cycle. Not included po si PPP, hence our answer is letter C for item number 59. Alright, so let's proceed with our next question. This is a hormone responsible for increasing gluconeogenesis in the liver during prolonged starvation. Class, when your gluconeogenesis is activated, meaning we need to produce glucose. So sir, meaning glucose is gone? Yes po. And clearly you know the answer, class. Okay? So again, when there's glucose is gone, we need to increase gluconeogenesis to replenish our blood glucose concentration and for energy generation. But before that, let's just discuss the functions of each hormones listed here. Class, so TSH is your thyroid stimulating hormone. Okay? So this is produced by your pituitary gland that signals that the thyroid gland should release your thyroid hormones. Okay? So that's not, hindi siya directly related in our gluconeogenesis expression. Okay? Letter B, of course, the insulin, it will increase glucose uptake. So actually, gagamitin niya kasi glucose instead of producing or inducing the production of it. Alright? So of course, obviously, letter B is an incorrect answer. Letter C, thyroxine, of course, this will slightly increase your metabolic pathway but not the main GNG signal. So, well, in particular, vague na hormone to. So it can increase the activation of your different meta pathway and thus not specific for GNG. And lastly, glucagon is our answer here. It stimulates GNG during fasting, starvation, low-carb diet, or in general, when we have low blood glucose concentration. Diba? We have summarized it last in our discussion here. For glucagon, of course, it will activate your gluconeogenic enzyme. But it will inhibit your glycolytic enzymes such as your PFK1 and your pyruvate kinase. On the other hand, of course, the opposite is true for insulin. Diba remember, ini-inhibit the insulin ang gluconeogenic pathway, but it activates your glycolytic pathway. Ayan, class, you just need to really remember the summary. So again, for glucagon, increase ang GNG, decrease ang glycolysis. For insulin, increase ang glycolysis, but decrease ang GNG for increase increase glucose uptake. Okay, so again, our answer for number 60 is glucagon, letter D. And class, remember, during our discussion, we also discussed your sites of secretion ni glucagon and insulin. So before we proceed with our next item, so let's just recall your pancreatic cells, diba? Here, class, I would like to emphasize the sites of your secretion ni glucagon and insulin. Again, for glucagon, your responsible site is the pancreatic alpha cells of islet of Langerhans. And of course, insulin, the beta cells, counterpart of the similar structure, your islet of Langerhans. So again, class, we have a mnemonics here. Ariana Grande, bad influence, diba? So I hope you still remember it. So for our item 61 kasi, you can directly use that mnemonics. Okay? So let's read. Beta cells of islets of Langerhans produce blank, which indicates blank blood glucose level. Alpha cells of islets of Langerhans produce blank, which indicates blank blood glucose level. Let's check. Again, for beta cells, Ariana Grande bad influence, that should be insulin. Okay? So sir, wala na po si letter C and D? Yes po. Alright, so now we are left only with letter A and B. Let's check the next blank. So beta cells of islets of Langerhans produce insulin. So when we have insulin, it indicates high or low blood glucose rather. So of course, diba, insulin for glucose uptake, meaning we have high blood glucose level. That's why here, wrong si letter B. And here our answer is letter A. Of course, alpha cells of islets of Langerhans will produce glucagon, which indicates low blood glucose level. All clear? Again, class, remember, beta cells insulin, alpha cells your glucagon and your insulin will indicate high blood glucose level, while your glucagon will indicate low blood glucose level. All right, for 61 again, our answer is letter A. All right, so class, let's proceed to our next item. Dagdagan natin ang correct answers nyo. All right, so number 62, ketone bodies serve as fuel for blank. Or this topic is under your lipid metabolism, particularly in ketogenesis. So again, our choices: A, extrahepatic tissues; B, hepatic tissues. Or class, remember, if you have this type of choices wherein they are almost similar, but again they are contradicting each other, either one of them is correct. Okay, letter C, erythrocytes, or letter D, mitochondria. Class, hepatic tissues are not the correct answer. Okay? So again, si hepatic tissues, site po siya ng ketone bodies formation. But our liver does not use your ketone bodies. O sir, ano nga po ulit yung reason? Because they lack the enzyme that catalyzes the production or utilization of your ketone body, particularly your beta-ketoacyl transferase. Diba? So I hope you do remember that specific enzyme. So here our answer actually is extrahepatic tissues. Again, your ketone bodies are produced by the liver, but used as fuel by extrahepatic tissues, since the liver lacks the enzyme to utilize them. Okay, so class, again, si erythrocytes and mitochondria, these are vague options. Alright, so of course, we are on a tissular level, not on your organelle and cellular level. Clear? For 62, our answer is letter A, extrahepatic tissues. Alright, let's proceed with our item 63. Ay sir, ang dali niyan, wala pang 5 seconds, nasagutan ko na. True? So let's check. What is the central metal ion of chlorophyll? Class, all of our choices here are all metal ions. So that's why you really need to remember the center or the central metal ion of chlorophyll. Ay sir, sorry, yung guardian angel ko yung nag-answer. I hope na tama ang answer ng guardian angel mo. Class, if you answered iron 2 plus and iron 3 plus, that's incorrect, okay? So this is the structure of your chlorophyll, and in its center, ayan, ay sir, magnesium 2 plus, parang sa kinase. Yes po. So our answer for 63 is letter C, magnesium 2 plus. Class, please remember again the structure of your chlorophyll. Again, it has a magnesium central metal ion in its structure, okay? 63, our answer is letter C, magnesium 2 plus. All clear? Alright, so let's proceed with our item number 64. So this is under your lipid digestion, di ba? So sodium taurocholate and sodium glycocholate are your bile salts. And they can emulsify fats because these two are A. Lipophilic, B. Hydrophilic, C. Hydrophobic, or D. Amphipathic. Class, these two are types of lipids. And we know your lipids, in terms of their structure, they are unique because again, they are amphipathic, diba? As we have discussed on our structure ng lipids. Class, in order to confirm, check nyo ang structure ng two bile salts. So of course, ang hydrophobic structure niya are the rings, of course, the steroidal rings, and of course, your hydrophilic component are your sulfate and amino and carbonyl groups. Okay? So that's why our answer is amphipathic. Sorry, class. So again, hydrophobic, the steroid ring, hydrophilic, the sulfonate, or taurocholate. So as you can see, ayan. And for hydrophilic naman, ng glycocholate, carboxy, and your amino group. Okay, so class, I do hope you remember the structure of your taurocholate and glycocholate bile salts. Alright, for number 64, answer is letter D, amphipathic. On number 65, the DNA in a prokaryotic cell is contained in a central area of the cell that we call your blank. So again, walang nucleus ang prokaryotic cells, class. Again, because wala siyang nucleus, obviously wala din siyang nucleolus. Sir, what is nucleolus po? That's the innermost structure of your nucleus where your DNA for eukaryotic cells are stored. Okay? So sir, letter D po ba ang answer? No po. Again, wala siyang true nucleus but it has your nucleoid in its cytosol. Okay? So again, class, remember our discussion in DNA organization for prokaryotic cell, it's stored in your nucleoid. Okay, so class, let's proceed with our item number 66. So this is still about your central dogma. So 66, the unwound strands of DNA are held apart by blank. Sir, parang nasa DNA replication yan. Yes po. And here are the choices. Is it A, single strand binding protein? B, double strand binding protein? Sir, parang wala namang DSBP. Yes po, obviously, letter B is an incorrect answer. Letter C, DNA gyrase or topoisomerase. Sir, no, that is to relieve supercoiling, not unwound strands. Yes. So this is an incorrect answer as well. And DNA ligase, class, again, this is to seal the gap or the nicks in your lagging strand. And obviously, our answer here is letter A, your single strand binding protein. Or class, check again. This is in the initiation step of our DNA replication. Clear? Ayan, again, for number 66, our answer is letter A, single strand binding protein or SSBP. Okay, number 67, class. Ayan, still under DNA replication, what type of synthesis occurs on the leading strand during DNA replication? Is it A, conservative, B, dispersive, C, continuous, and D, discontinuous? Again, class, in terms of the synthesis, we can already eliminate conservative and dispersive. Okay? So when we say conservative kasi, this is incorrect. Because even in our replication, semi-conservative po tayo, di ba? One parent strand or all strand plus one use strand ang product natin. When we say conservative, meaning your entire original DNA stays intact. So this is not how DNA replicates in cells. So sir, how about dispersive? So incorrect din siya because again, your dispersive replication would mix old and new DNA segments throughout both strands. So meaning, nag-mix and match a class in one uniform strand or in one single strand. So between continuous and discontinuous, of course, we are just particular to leading strand. In the leading strand, ayan, it is continuous. But in lagging strand, discontinuous. So class, again, we are asked about the leading strand synthesis and thus our answer is continuous. Okay? So I hope it's clear for number 67. Our answer is letter C, continuous. Class, again, kamusta ang scores? Sir, pumasa na. Wow, wala pa tayo sa last number, pumasa na. Good job, class. So in order to add up on your correct answers, let's proceed with our next item. The origin of replication starts at regions in the DNA that blank. Class, remember, I emphasize this, diba? So our answer here must be letter A. Again, your AT base pairs compared to your GC base pairs, relatively weak ang interactions because it is only held by 2 hydrogen bonds compared to your GC that have 3 hydrogen bonds. Okay? Sir, how about the hairpin arrangement? Class, this arrangement are for secondary structures formed by your RNA, not in your replication ng DNA. Okay? For your supercoiling, of course, this is the site of your topoisomerases reaction. Remember, class, preparation ng elongation step, not in your origin of replication. Okay? And then letter D, are already single-stranded? No. Again, your SSBPs and your helicase help your initiation complex in unwinding your DNA double helix. So class, for 68, our answer is letter A. Again, the ORI site or origin of replication begins at AT-rich regions because your AT base pairs again have only 2 H bonds, making them easier to unwind than your GC-rich regions. Again, for 68, answer should be letter A, are rich in AT base pairs. Alright, so class, we are now on our next item. 69, medyo malapit na tayo, no? So for number 69, 2 types of adapter molecules that are required for translation are the, class, remember, I emphasize this, diba? So your adapter molecules, they are concerned in your tRNAs. Okay? So letter B. Sir, letter A ang answer, in-skip mo. Yes, but let's just discuss first letter B. Alright, so tRNAs and introns. So class, remember, introns are not adapter molecules, diba? They are non-coding RNA sequences that are removed during RNA processing and they are not directly involved in translation. So your letter B is incorrect. Letter C, amino-acyl tRNA synthetases and SNRPs or SNRNPs. Tama po si amino-acyl tRNA synthetases, but si SNRNPs, again, involved po sila sa RNA splicing and not in your translation process. And of course, introns again, class, it's incorrect. And clearly, our answer is a tRNA, which will carry your amino acid, diba? And your amino-acyl tRNA synthetases. So that's a requirement in order to charge our tRNA with the correct amino acid. So clearly, for 69, our answer is letter A, tRNAs and amino-acyl tRNA synthetase. Again, si tRNA, amino acid carrier to the ribosome, and amino-acyl tRNA synthetases will charge your tRNA. Class, again, remember our discussion here. Please revisit this. So this is on your tRNA activation. Remember, we used ATP pa nga here in order to activate your tRNA. Or remember when I say tRNA, it's just adding your amino acid in its 3' to 5' CCA. And alright, so class, that's it for our item number 69, letter A. Let's proceed with our next item. Number 70, sigma factor is important for blank. Class, before I discuss, here are the choices. So as you can see here, we discussed this during our discussion on transcription, right? So particularly, sigma factor is important for transcription initiation, specifically for your prokaryotic cells. I hope it's clear again. Our discussion here involves the assembly of your initiation complex in our first step of transcription. Primarily, RNA pol, with the help of your sigma factor, will bind to our promoter sequence in order to initiate the assembly process for the start of transcription. So as a summary, the sigma factor helps RNA polymerase recognize and bind to the promoter during transcription initiation in prokaryotes. So clearly class, answer for 70 is letter D, transcription initiation in prokaryotes. Let's go to our next item. Ayan, so this is about the repair mechanism in our DNA replication. So DNA photolyase recognizes which of the following in order to repair pyrimidine dimers. Ayan, is it A, distortion in the double helix, B, a specific palindromic sequence, C, a specific origin for repair to initiate, or D, 3H bands between affected base pairs? Let's discuss. For letter D, this is an incorrect question. So class, your pyrimidine dimers involve thymine or cytosine. So they normally have 2 hydrogen bonds, not 3, okay? So again, the dimer formation disrupts normal hydrogen bonding. Again, it's not 3 hydrogen bonds. Rather, for pyrimidines, we have 2 H bonds. Letter C, a specific origin for repair to initiate. Class, repair enzymes don't need a defined origin like replication does. Okay? So again, letter C is an incorrect answer. Hindi nila need ng mga oocytes. Alright? Sir, letter B, correct po ba yan? No po. Okay? Your DNA photolyase actually is structure specific and not specific sa sequence. Okay? So clearly, si letter B is incorrect. Sir, letter A ba ang answer? Yes po. Again, class, ang pyrimidine dimers kasi is caused by your UV light, diba? And of course, it will induce your pyrimidine dimerization, and that's a distortion in your double helix. And that's the mechanism of DNA photolyase. It recognizes those distortion or bulky structure in your DNA structure. Alright, so again, class, this is the specific diagram. So your DNA photolyase will identify pyrimidine dimers by detecting the distortion or the damage in your DNA helix. So again, hindi siya sequence specific or bond specific. Alright, so class, let's proceed with our item number 72. Ayan. So which amino acids are used in human de novo purine biosynthesis? Class, I hope you remembered our brief discussion on purine de novo biosynthesis. Is it A, lysine, aspartate, and glutamine? Sir, sorry, di ko alam yung 3-letter codes. Class, it's okay, may time pa kayo to memorize your 3-letter codes for amino acid. Going back, letter B, aspartate and glutamine. Letter C, proline, aspartate, and glutamine. Or letter D, proline and glutamine. Class, never magiging part si proline ng ating purine biosynthesis because of its structure, okay? So now, it's just either A or letter B. O, ibibigay ko na yung sagot. I will splook it na. Ayan, letter A, ang answer natin. Remember our discussion on purine biosynthesis. Ayan, I put it on bold pa nga in order to emphasize it that your lysine, glutamine, and aspartate are sources of your amino acids or nitrogens in your purine biosynthesis. Okay? So class, I hope it's clear. Again, for our item 72, our answer is letter A, your lysine, aspartate, and glutamine. Alright, so for our next item, class, konti na lang. So I do hope at this point, nasa passing score levels na kayo because we only have 8 answers left to discuss. Alright? So going back. Which amino acid has only one codon? O sir, sure ako may methionine. Class, lahat ng choices may methionine. O sir, guardian angel na lang, hindi ko naaral eh. O let's check. Again, this is your tabular genetic code. And class, I have already extracted your amino acid and their corresponding number of codons. Okay? So again, here is our list of codons. So let's check. Ayan. So class, obviously our answer is methionine and your tryptophan. O check nyo sa tabular genetic code. For your methionine, AUG only. For your tryptophan, UGG only. Alright, so class, again for 73, letter C, methionine and tryptophan. Sure, mali ako, hindi ko naaral. It's okay, class. At least, right now, you now know the answer. Alright, so again for 73, letter C, methionine and tryptophan. Alright, so item number 74. Which amino acid is not glucogenic? Meaning ketogenic siya, sir? Yes po. O diba, we only have 2 amino acids that are exclusively ketogenic or ketogenic, diba? That's your LL. It's either your leucine or lysine. O sir, si leucine lang yung nasa choices. Is that the correct answer? Yes po. Okay? So again, they are not glucogenic. They are exclusively ketogenic. So for number 74 class, again, remember, we only have 2 exclusively ketogenic amino acid, si leucine and si lysine. Okay, so again, for 74, A, leucine. Now let's proceed with our item number 75. What is the tRNA anticodon sequence of the starting codon? O class, remember, our starting codon is 5' AUG 3', diba? So this is your starting codon. So hindi siya tRNA anticodon. So wrong na yan, class. Ang answer dyan dapat is complementary with our starting codon. Okay? So if this is our starting codon, dapat here is the 3'. Again, dapat complementary sila. So dapat to 3' and then paired with A, CU in your RNA, and then with U, CA, and then lastly with G, CC, and then complementary with 3' is 5'. Sir, si A ba ang answer? Yes po. Okay? So si letter C, class, this is a correct sequence as you can see in its nucleotide or the bases, but in wrong direction, 5' to 3' siya. Alright? O sir, si letter D, class, incorrect sequence. Alright? It's the same as the mRNA codon, but again, wrong ang direction. Okay, so again, for 75, our answer is letter A, since the tRNA anticodon must be complementary and antiparallel to the mRNA start codon. So again, remember, 3' UAC, 5'. Alright, so class, we are now in our last crash course topics under your lab techniques. Okay, so I do hope may na-answer kayo. So number 76. SDS page is used in what manner? Class, remember, ang ating charge or net charge or charge to mass ratio in your SDS is uniform. Hello sir, meaning mali si A and B? Yes po. So class, again, in your SDS page, our basis of separation ay molecular weight. Okay? And here in particular, in our SDS page, is in decreasing manner. Okay? In your SDS page, much faster ang migration ng low molecular weight proteins compared to the larger ones. That's why, again, our answer here is letter C because our SDS page separates proteins based on size and clearly from largest to smallest. Plus, diba remember, we discussed pa nga in detail your agarose gel electrophoresis. So, of course, we have our reagent or different reagents that we need in order to do your age or SDS page in particular. All right? So, of course, class, remember, in our visualization, we can do this via staining or through blotting techniques. All right, so class, let's proceed with our next item. Number 77. Which of the following is a reducing agent that disrupts disulfide bonds during SDS page? Or class, check the choices. Letter A, SDS. No. Again, this is to the nature of your proteins. Letter B, acrylamide. Sir, hindi ba gel matrix yan? Yes po. So again, it is not a reducing agent. Letter C, letter D muna class, di ba? Letter D, Comasi blue is actually a staining, di ba? For staining or for visualization. And clearly, our answer here is beta-mercaptoethanol. Okay? So again, it breaks your disulfide bonds by providing an alternative site of interaction or reaction in its thiol functional group. Okay? So class, for 77, our answer is letter C, beta-mercaptoethanol. Alright, so for our next item, ayan. Sir, challenging yan. Na-challenge kami. Let's discuss this in detail. Of course, this is an analysis type of your Chrome experiment. So between beta amylase with an isoelectric point of 5.17 and cytochrome C with an isoelectric point of 9.20, they are dissolved in the buffer of pH 7 and loaded on a cation exchange column, which will elute first. Class, familiar ba sa cation exchange column? So I do hope that you are familiar again in your cation exchange column. This binds your positively charged or cationic proteins. That's why from the name itself, it binds your cations. All right. So here are the choices. A, beta amylase. B, both proteins will elute at the same time. C, cytochrome C. Or D, both proteins will be bound to the resin. Class, I will give the answer first. Nanghula ba kayo? Or nag-try mag-analyze? So I do hope you tried analyzing the problem. But in this item, our answer is letter A, beta amylase. Alright, so let's discuss this in detail. So again, your cation exchange column or your resin binds positively charged cationic protein. So meaning, matitira siya class sa column natin, okay? Before elution. So let's check. So at pH 7, recall si beta amylase, ang PI niya is 5.17. So clearly, it is below our current pH of 7, di ba? So that's why pH is greater than PI and then meaning may net negative charge po si beta amylase. Okay? And that's the reason why it won't bind. It will elute early. Okay? So on the other hand, si cytochrome C, ayan 9.20. So of course, class, it will carry a net positive charge because again, it's your pH is less than the PI. Alright? So again, hindi pa niya na-reach ang isoelectric point. Remember, if your pH is greater than PI, of course, deprotonated na po ang ating amino acid or the enzyme in particular. Now, kapag si pH is less than your PI, of course, protonated pa rin po. Di ba we discussed this in our discussion on your isoelectric point. That's why for cytochrome C, it will still carry a net positive charge and thus it will bind to your resin. That's why siya ang mag-elute na last. Okay? So class, again, our rationalization is here. For beta amylase, it's negatively charged at pH 7. That's why it will elute first because hindi siya magbabind at all. Okay? So cytochrome C, ayan, it will bind because it has a net positive charge. Okay, so class, yung both na answers, of course, impossible yan because they have different PI values. Alright? So for our item number 78, again, our answer is letter A, beta amylase. Alright, so class, I hope you remember our discussion on isoelectric point. Remember, when pH is greater than the PI, net negative charge. When your pH is less than your PI, ayan, net positive charge na po, meaning it's still protonated. So let's proceed with our second to the last item. Excited na kayo? We are almost done. 79. Edmond degradation is a method of sequencing amino acids in a peptide, and in this method, the N-terminal amino group is reacted with which reagent? Okay? So class, si 2,4-D and FB, si phenyl isothiocyanate, and si densyl chloride, they can all attach or react with your N-terminal amino group. But here in this question, we are particular with Edman degradation. Now, di ba recall in our protein classical analysis, ayan, your phenyl isothiocyanate is our reagent being used for the N-terminal amino group structure elucidation. Or class, baka nagka-amnesia, again, in our discussion, this is the table that I have presented, and here we use PTC or phenyl isothiocyanate for Edman degradation. Alright, and then last item, sir, tama ka na. Answer all of the above. Yes, of course, our answer as a characteristic of a good cloning vector. Class, I give you a summary of a good cloning vector. Let's cross-check. Letter A, it has a restriction site. Yes, correct. B, it is small in terms of size. Yes, correct. Letter C, it has a selectable marker. That's why answer is all of the above. So class, again, please review our characteristics of good cloning vector. And that's it for our discussion of your evaluation exam for biochem. Class, I do hope that majority of you got a passing score. Okay? So again, our passing score is 56 out of 80 items. Class, for the summary of our answer key, I will flash it for 5 seconds per slide. Okay? Ayan, again, for 80, letter D, all of the above. So this is for 1 to 20 class. So you can pause the video. This is for our 21 to 40 items. For 41 to 60. And then finally, class, this is for our 61 to 80. Okay? So please check again your answers. And then if you have questions, class, you can consult me during our final coaching session. Alright, so class, that's it for our EA discussion for biochem. I do hope that you are reviewing and studying smart. Alright, so class, bye. Enjoy the rest of the day.